talk to you about a project that we've been doing the last few years where we've been looking at minimal genomes and focusing on inferring the functions of the essential genes of life. So if we think of the area of synthetic biology, there's been a number of quests, and one of those has been to identify the minimal genome, i.e. the species with the smallest possible genome, such that the cell or the species is able to divide and replicate and effectively live. At the same time, there's been another quest to generate synthetic genomes as well. So a lot of this research has kind of come together, and this slide here gives some of the history that we've seen. Now, a lot of this is from Craig Venter's group, and if we see 95, there was a minimal gene complement for Mycoplasma genitalium. So this is one of the smallest bacterial species. And then move on a few years later, and they've used global transposon mutagenesis to look at minimal Mycoplasma genomes. And there's more work in 2005, and there's a lot of other papers as well. Now, we move forward a few years to 2016, and there was a paper in Science, Design and Synthesis of a Minimal Bacterial Genome. And so this was the first paper where we had a truly synthetic genome, and it was also the smallest genome for any species that we've sequenced or identified to date. And this paper, the research in it, is really what has driven the work we've done in some of the work we've done in our group for the last few years. So how did we get interested in this? Well, I happened to be going to a conference in Copenhagen in early April 2016, and I was in the bar of my hotel the night before the meeting started, having a drink, and I noticed among the magazines they had there, they had a copy of Nature. So I had to flick through, and there was a News and Views article, and it was about this recent minimal genome paper. And given my interest in protein function, I was quite fascinated by it. And here's a brief excerpt from that News and Views item. And clearly here, Craig Venter is highlighting that there were 149 genes in this genome, which is called SYN 3.0. And that was about a third of the minimal genome that they'd created or generated, and they didn't know their functions. So it's a third of the essential functions of life, and we don't know what they are. And given my background, so I've had a lot of roles in development of computational methods. So ComFunc is a method we've developed for inferring gene ontology functions. 3D ligand site, a method for modeling small ligand binding sites in proteins. And I've also been involved in FIRE2, protein structure prediction tool led by Mike Sternberg at Imperial College. So given this background, I was thinking, well, there's lots of tools, lots of computational bioinformatics tools that we could use to try and learn more about these proteins. And that if we think about that in a broader context, it's also been involved in all of the critical assessment of functional annotation experiments. At that time, we'd been involved in the first two with the papers were published here. We'd done quite well in those methods. And clearly we were aware that there were lots of other groups as well developing methods that could be used to do this. So a few more words about the synthetic minimal genome. So it's based on a Mycoplasma mycoides subspecies. And this is a picture of the cells growing. It contains only 473 genes, of which 438 are protein encoding, the rest encode RNAs. And at that time, from the science paper, 149 of those protein coding genes were classed as having unknown function. So in that paper, they assessed or assigned functions using mappings to TGRAPHAM. They used genome context, and they used protein structural modeling using a threading approach. So they used three different methods to come up with the annotations that they assigned to the proteins. And they did this in two different ways. So they had functional categories which we can think of as being like a biological process. And then they had a functional class for each gene or protein. And this strangely seemed to encapsulate an element of molecular function, but also confidence in the overall annotations as well. And those functional classes, there were 5 of them: unknown, generic, putative, probable, and equivalog. And we see here the number of genes in each of those groups. And equivalog was where there's clearly an ortholog, known function in many other species. And then probable and putative, there's some match that we're fairly confident in. And then generic and unknown were groups where there was really, as the name unknown suggests, no functional information identified. And in generic, some basic information was available. And if we look at the number of genes in each of these groups, we can see the equivalog group has close to 200, and that might not be surprising. A minimal genome is probably going to include lots, well, it's all of those genes that are essential for life, things to do with duplicating DNA, gene expression, and so on. And so we've studied many of those proteins across life sciences, and so lots of those have annotations. But what this group here, these two groups, 149 proteins, and these are really the ones with unknown function that we were very much interested in seeing if we could learn more about them using bioinformatics approaches. So before we move on to thinking about predicting the function, we wanted to think about what the properties of these proteins were. So we performed some analyses. So we started off by looking at orthologs, and we used eggnog to do this. And we can see that in the unknown group, then there tended to be very few orthologs across the 3 kingdoms of life. And there's a few more in the generic, but increasingly more when we go up into the putative, probable, and equivalog groups. Then we were thinking about protein domains. So what we found, we have in the unknown group, very few proteins where we can identify domains present in PFAM. Most of them, we don't get any matches. There's a few with a single domain, and there was one entry, I think, which had multiple domains in it. And then if we clearly see a big difference then when we go to the generic group. So we've got some basic information about function here, and we can see that most of those proteins now have a match to a domain. For most of those, that's a single domain, but there are some with multiple domains. And if we contrast that then with the equivalog group, those where we have the most confident functional annotations, we see that they've all got matches to PFAM domains, and nearly half of them have multiple domains. So then we're thinking about disorder. So again, we saw the unknown group seems to have slightly higher proportion of proteins that contain disorder, but it's not a huge difference here between any of the groups. Now, given our interest in modeling protein structure, we use the FIRE2 method to investigate what proportion of each protein we were able to model. So plotted here, we're indicating the percentage of each protein sequence that we are able to confidently model using FIRE2. And we see that with the unknown group, there are a few proteins that where we can model all of the protein sequence, but for most of them, we were not able to identify a template that we could use for modeling. And again, we move to the generic group, we're able to model many more of the proteins. And then for the other groups, we can see most of the protein sequences, we can model nearly all of them. Then we thought about transmembrane proteins. So we noticed that quite interestingly, these proteins in the unknown and generic groups seem to be enriched in transmembrane proteins. So nearly 50% of the proteins in the unknown group have transmembrane helices. A considerable proportion of the generic proteins in the generic group as well. And then if we contrast that with the other three groups, particularly the equivalent group, we can see that most of these proteins don't have transmembrane helices in them. Then if we delve a bit more into that and look at the number of transmembrane helices that are predicted, we see that very few of them have a single transmembrane helix. Many of them have two or more. Particularly in this generic group, we're seeing that most of them have 6 or more transmembrane helices. So that's something that we'll come back to later on. So that's just summarizing this first part. So we've seen that these proteins of unknown function, they seem to be depleted in orthologs, depleted in the presence of known protein domains. They are enriched, possibly for disorder. They're enriched in transmembrane helices. And we also lack structural templates to model the protein structure. So that maybe explains some of why they've been difficult to annotate, because there doesn't seem to be a huge amount of information available about them. So having, having done that, we wanted to move on now to think about, can we infer the function for any of these 149 proteins using the many computational methods that are available? So we used a combination of 22 different approaches. Many of them are listed here. So we had domain/ortholog type methods, used protein structure. We used 2 methods for binding site predictions. We used a number of methods that related to membranes and transporters. And then we used a number of gene ontology-based methods that are generally predicting protein function. So I recognize there's lots of different methods available, and I'm sorry if we didn't use yours, because we can't use all of the methods that are available. Yes, I've seen someone comment about genome context. Yes, we did look at genome context. It was in there somewhere. But yeah, it's not going to appear in any of my slides. So that's what we did. Now, here's a figure from the paper that summarizes for one of the proteins how we went about that. And I think it's kind of easier if we simplify it into that we had domain and orthology methods, we had structural methods, we had methods for binding sites, GoTerm-based methods, and methods relating to protein membranes. And so, because we had a small number of proteins here, what we could have done is made some sort of machine learning classifier that would have taken information from all the different resources. But given that we had a small number of proteins, I wanted us to try to combine the information together manually so that we weren't relying on a machine learning tool to do it for us. And it's quite a tractable problem with only 149 proteins to deal with. So this was largely done by my PhD student, Magda Antchak. And so she would take the methods from the domain and orthology methods and the structural information, and that would give us some initial idea about the function. And then she would combine this with the results from the GoTerm methods and the membrane-based methods and the binding sites to give us a final overall function that we have inferred. And then we assigned a confidence score to each of those. And what we did was we took the scores from all of the different methods and we normalized them so that they each ranged from 0 to 100. And then we averaged them. So we got a final average score here. And we also recorded the number of methods that had been used to provide that function. So what we wanted to do was infer the biological category or the biological process and also molecular function. And now we found these functional classes, the equivalogs, the generic, the unknown, a bit difficult to use because they, as I said earlier, seem to encapsulate elements of molecular function, but also a level of the confidence of the annotations that we had for the protein. So what we tried to do is split those out. So we came up with specificity categories that indicate how specific the function that we've assigned is. And that starts at hypothetical, where the function we don't seem to know at all. We haven't got any information. General, where there's some basic information. So for example, this might be that a protein is a transporter. Specific, where we've got some more information about that function. So this might be that that transporter is an ABC transporter. And then we had highly specific, where we've got a higher level of detail again. And now, if again with that same example, this transporter, we might then know it's a substrate. And as I was saying in the previous slide, we've got a confidence score ranging from 0 to 100 that combines all of those individual methods. So with anything, we didn't want us to run off and to use it. We wanted to do some benchmarking, and so we tested it on proteins in the minimal genome where we already knew their function. So that's these three groups: the putative, the probable, and the equivalogs that we saw from those earlier categorizations. And we applied our approach, and we compared the functions that we got to the functions that were present in the original science publication. And thankfully, we found that the annotations agreed for 92% of the proteins. There were a few mismatches here. Sometimes one was more specific than the other, and there was only a single protein where we disagreed on functions. So that seemed to be a good start. So now I'm going to move on and think about those two groups that we had, the proteins in the unknown and the generic groups, and what we found out for those. So of the 149 proteins in those two categories, we improved the annotations for 66 of the proteins. And for 51 of those, we increased the specificity class. I'll go into that in a bit more detail. So here we have an image that indicates the initial class that proteins belong to. So we had our hypothetical class, the general class, and the specific class. And then the color of the methods indicates whether, so these are all the different methods that we used. And the color here in the boxes indicates whether we had information for that method and whether it supported the function that we came to. So, and then the color indicates the specificity class it went to as well. So what we see that for many of the proteins where we initially had a hypothetical class, we've moved many of them to a general function. And then a few have moved to a specific function, typically where there was more functional information available for more methods. And then one of them we've moved to a highly specific function. I've highlighted these two here because I'm going to show you examples of those in a moment. And then proteins that were initially in the general class, well, there weren't that many of them that we've then annotated, and those have all moved then into having a specific function. And we can see that there's data available from increasingly more and more methods. And then finally, we had a number of proteins that were initially in a specific class, and we've been able to fine-tune and add more detail to those functions that they're now classed as being highly specific. And again, that's helped by having functional information available from the majority of the methods that we've considered. Now, if we just go back to this hypothetical class for a moment, we actually see that for most of the proteins, there were really only three methods that made predictions. So that was the combination of the methods from gene ontology terms, the method TRSSP that predicts transporters and their substrates, and we were also obviously for all proteins able to predict whether or not they have transmembrane helices. So it clearly indicates that there was limited functional information available still for a proportion of the proteins. But even so, we were still able to add more functional information than the lack of information that was initially available. So if we think about those functions, and we can now think about the biological process or the functional categories that they belong to. So in this graph here, I'm showing each of these categories. So translation has got the most proteins or genes in it. Then we have this unclear category where, as the name suggests, we're not sure what the function is. And we can see that initially, so all of the lighter colors show the numbers initially, and the darker colors the number after our analysis. And clearly, we've had a significant reduction in the numbers of proteins in this category. You see a few added to translation. There's a large increase in the number of proteins that are now appear to be associated with transport. And we can see there's a few other changes in some of these other categories as well. So overall, when we look at this minimal genome, we see that more than 50% of it is responsible for preserving and expressing genomic information. And if we look at our specificity classes, we can see that initially there were a number in the hypothetical class. Now there's far fewer in that hypothetical class, and those have largely then moved to the general or the specific classes. So I want to use a few examples to show what we found. So this protein MMSyn 298, it was classed as being a hypothetical protein initially, and we've got data from multiple methods. So we had Fire2 with a structural model, more structural information from Cath FunFams, and also functions as well there. Results from domain-based methods. The gene ontology term methods supported the function as well, and also from EGMOL. So we've ended up with a prediction of this being a ribosomal protein L7AE L30E family. We look at a second example, again, another protein that was classed as being hypothetical. And again, we've got structural information. We had a binding site predicted as well. Lots of confident predictions from the gene ontology-based methods and also from the domain-based methods. And so we've ended up with a prediction that this protein is an oxygen-sensitive NADPH nitroreductase. And so now I'm going to move on and think a bit about the transporters that we noted seem to be enriched in this minimal genome. So just one example here to start with, this protein MMSyn 325, and it was identified as being a putative membrane protein by the initial study, and it had some generic level of confidence. So if we start out here, we had a clear prediction of transmembrane helices from TMHMM, so it's clearly got transmembrane helices, 13 of them. From Fire2, we were able to generate a highly confident structural model showing all of those transmembrane helices, and there were 26 templates, and all of those had transporter and permease functions. Then when we looked at the Go term functions, we had many that were highly confident associated with transporter functions from ComFunc and FFPred, including Symporter activity and ion transmembrane transporter activity. We also had from Interpro, this was an MFS transporter superfamily, and I should add the templates were also from the same superfamily as well. So now we have then predicted that this protein is likely to be a cation transporter and a member of the major facilitator superfamily. So overall, we saw that there was this large increase in transporter functions. And there were 23 new proteins that we annotated with transporter functions. And they ranged from specific, so we identified a hexose phosphate transporter, an ABC transporter, an S component of an ECF transporter, 3 proteins that are part of the major facilitator superfamily. And then there were 18 proteins that we've more generally labeled as transporters, but we have less functional information available about them. So overall, across the minimal genome, there are just over 60 transmembrane proteins, and 46 of those are very likely to be transporters. So clearly, it seems that this minimal genome is enriched in transporters. And so now I want to think about why that might be. So this got us thinking about what a minimal genome is. Now, what I should have set out at the beginning and explained is that this minimal genome is grown in a nutrient-rich medium. So that's led us to this idea that a minimal genome contains 2 sets of genes. So there's this first set of genes that are truly essential for all life in all species, and they're shared by all the different species that we have. And then secondly, there are genes that are essential depending on the environment. So our minimal genome that we're investigating is present and grown in a nutrient-rich medium. And so that means that you can, or the cell can import many nutrients and export waste products. And so genes that would be needed for metabolic purposes, many of those can be removed because you're relying upon transporting in everything that cell needs from the external environment. So then if you were to look at a minimal medium, now this is just a basic diagram. I'm not trying to suggest anything specific here about the number of genes required in a given minimal genome in a given medium. But if we go down to a minimal medium, we've got far fewer nutrients. And so then this minimal genome, the cells are going to be relying upon those metabolic pathways present within the genome to be able to generate those products that they need for life. So clearly, by growing the Mycoplasma mycoides synthetic genome in a nutrient-rich medium, it's minimized the size of the genome. And that, at the same time, has then made these transporters essential. So if we had a minimal medium, those transporters might not be essential because there aren't nutrients that they're able to transport into the cell, and instead the cell is relying upon metabolic pathways instead. So what does our study tell us about protein function prediction? So something that became very obvious when we were doing this, and something I think we've known for some time, is that methods are largely reliant upon homology. So there's literally only 1 or 2 methods that we used, I think of FFPRED, for example, that aren't based on homology information. So clearly, if we want to be able to predict the function of more difficult proteins, we need more methods that are not homology driven. Someone in the chat has already mentioned that maybe we could redo the modeling with AlphaFold. So yes, we've now seen in the last year huge advances from AlphaFold, and I'm sure there'll be many other methods for accurate modeling of protein structures. And clearly that now offers the field of protein function prediction the potential for developing many structure-based methods, because we're now going to have confident structural models available for nearly all proteins. And we're not limited for structural methods to a small proportion that are present in the protein data bank, or for which we can generate highly confident models or were in the past. So I want to briefly mention some analysis by another group, and that's David Jones's group at UCL. And that builds upon this last point about using the latest methods. So 2019, David Jones's group published DMP-fold, deep learning-based method for protein structure modeling. And so what they've done is they've taken the same minimal genome and run it through DMP-fold. There's a preprint of this available on arXiv, and they've been able to model fairly confidently structures for all but 10 of the minimal genome proteins. And my understanding that even for those 10 where they're not very confident of the models, that's because while they have a model, that model doesn't really match existing entries in the protein data bank, which is a way of getting more confidence about the model that has been generated. And they've then used similarity of the DMP-fold structures or models that they've generated for the proteins to map them against the PDB to propose potential functions. So here we have an example, MMSyn 375. Here we show the model and the closest structure that they identified. And this is a clear structural similarity to an ATPase. And so that's a protein that we started off with where we weren't able to infer any function. So clearly, we need to make use of the latest methods. We tried that when we were doing our approach. And it just shows how quickly other new methods come along. So that's all I wanted to say today. Just to summarize, we've used a whole range of bioinformatics methods to expand our understanding of the function of a minimal genome. Through that, we've seen that a minimal genome depends upon the environment that it grows in. So we saw this need for transporters because had a minimal genome growing in a nutrient-rich medium. And then we've also seen that we need to use methods that don't just rely upon homology, and also, as a field, we need to make use of the latest advances in protein structural modeling. So thank you for your attention, and I'll pass over to Ido. I think we have time for some questions.
Yes, thank you, Mark. That was a great talk. Really very thorough examination and upgrade of essential genes. So I guess we learned something that unknown essential genes. One of them that you found out was related to information transfer, right? So that will be the ribosomal protein, and the others were and there was general rich in transporters, which was interesting, given that this is a mycoplasma, and mycoplasma in general can only live in rich soups anyway. So that really makes a lot of sense. So there are a few suggestions here. I don't know, Seth, can we get people to actually talk, or does the audience only communicate through the chat in this medium?
Chat, but we have a small audience today, so I will invite him to the stage here real quick.
Oh, great. Okay, fantastic. Yeah, hopefully it will facilitate a more a freer dialogue rather than via chat. Hi, Gabo.
Hey, how are you?
Good.
Good.
So
Thanks.
you had a lot of comments. Maybe we should start with you while Seth elevates everybody else to the stage.
Yes, Mark, I quickly found the paper, and I saw that you did predict operons, but only in a few mycoplasmas. So what I'm suggesting is that you predict them in anywhere you find a homologue to your proteins, right? So that you build a network of potential interactions, and that might help.
Yeah, Yeah, we could definitely do something like that. Yes, thanks.
Yeah, sort of guilt by association.
Yeah,
if you
but
have
with
one homologous
a
anchor.
wider network, yes, because the operons rearrange, so they don't stay in a single operon in every organism. They move, and normally I have found that the networks that are built are consistent with known functions.
Right. Okay. Yeah. No, that's a really good suggestion. Thank you.
Yeah, but very interesting work. Thank you, Mark.
Okay. Any other questions from the audience, either in chat or speak?
Okay, then just can you tell us if you've been already doing some of the AlphaFold predictions?
We haven't actually, primarily because my student finished her PhD and she's gone off somewhere else, and haven't got anyone else currently to do the work.
So I think that EBI are slowly putting together an arsenal of AlphaFold predictions. I don't know if they even started on bacteria yet.
There's a database of model species, so
yeah,
Mycoplasma mycoides isn't one of those, I don't think.
that's a shame. Should maybe talk to them. So that sort of, obviously need to actually get the AlphaFold installed in the university cluster or something and start running it there, which can be a little bit painful from personal experience.
Yeah.
Yeah. So my comment is, I was just fascinated with the whole idea of essentiality varying according to where you live, right? So what is essential for a mycoplasma might not be essential for Bacillus subtilis. So a large survey of genomes and metagenomes for enrichments in various categories might be something to try and do. So I put my thoughts there in the chat. And,
yeah, definitely. That's a really interesting idea. It's not something we've explored, but yes, even within the same. So if you'd started with the same here, with the same mycoplasma genome initially, but you'd had two different environments, you'd have ended up with two different minimal genomes. If you had one that was in a low nutrient medium, you probably wouldn't have these transporters. You'd have more metabolic enzymes retained compared to what we have in this one that's been grown in this nutrient-rich medium.
Yeah, so we actually looked at mycoplasma specifically that infect swine, and generally there's a pretty big difference once you passage them and in the lab in terms of their genome, and also the same species picked from different farms seem to be quite different sometimes. So it's really interesting. Mycoplasma are incredible, I guess other bacteria as well, but these are parasitic intracellular organisms. So they're incredibly versatile in what they adopt and what they shed from their genome over a very small number of generations.
Yeah.
Are there any other questions or comments for Mark?
So I can see in the chat that R.J. Murray has commented. They've asked what makes AlphaFold the standard over the other options. So I can answer that quickly in that AlphaFold is this new deep learning method developed by DeepMind, and at the recent critical assessment of protein structure prediction, it was demonstrated to outperform all of the other existing methods, and quite significantly as well, to the extent that the models it generates for most proteins we have really very high confidence in.
Yeah, I might add here that this is a fairly recent determination. That's not even 2 years old, I believe. So it's slowly gaining acceptance, and there are a lot of various efforts now to AlphaFold, as it were. It's becoming a verb to AlphaFold whole genomes, and to fill in the gaps in terms of our knowledge and structure. That's a very, very interesting new horizon in terms of our understanding of sequence structure function connections. It is quite AlphaFold is quite impressive. If you look at the latest CASP charts, it really is outperforming by quite a bit, even the second method and so on. And not only outperforming according to the metrics of CASP, it's really performing very well. It's not only relative good performance, it's absolute good performance. Even for I think the hardest proteins to predict, it gets a score of 80 on a scale of 0 to 100 using the popular CASP metric for performance. And for those that are, supposedly easier to predict, it gets over 95 or something. So very good performance. Yeah, it probably already is a game changer.